Question number 136. Triticale. Triticale is an example of, we know the ploidy. The ploidy is a condition where the accumulation of the mass of cells. This question, triticale or triticale is an example of allopolyploidy. Allopolyploidy is the correct answer for question number 136. 136, allopolyploidy. Allopolyploidy is the correct answer for question number 136. So, option 2 is the correct answer for question number 136. Alloploidy, where we could see the polyploidy. Polyploidy is the phenomenon of having more than the two sets of chromosomes of genomes, but allopolyploidy is developed through the hybridization between two species, through hybridization between two species followed by doubling of the chromosomes. Triticale is the first man-made alloploidy developed by crossing breed, that is Triticum, Tergidium and rye. So, where you could see in this case triticale, the triticale which forms by the fusion of you could see the rye and a wheat. So, this is called it as triticale, which comes under allopolyploidy. This is the first man-made product. 136 is the 2 is the correct answer. 137. The semi-dwarf wheat, which was instrument in increasing the wheat production, was developed by Norman E. Borlaug, is considered as a father of a green revolution. So, father of green revolution, question number 137, Norman E. Borlaug. Okay, he is actually the father of green revolution. So, question number 137, 4 is the correct answer. Then question 138, the correct answer is biofortification. So, breeding of crops improve nutritional quality. We already discussed about this. Biofortification. Option 3 is the correct answer. So, what is the main objective? Main objective to improve the biofortification. This we already discussed already, right? We discussed biofortification to improve the protein content, then the quality, and to enhance the oil content of the quality, to increase the vitamin content, to improve the micronutrient and the macronutrient content, right? So, for this one we have the option 3, that is biofortification is the correct answer. Okay. So, 139, question number 139. Breeding of disease resistance requires a good source of resistance that we required. Plant hybridization is required. Disease test is also required. So, all of these is the correct answer for 139. Question number 140. Both assertion and reason are false for the question. The assertion is the technique. The assertion that you could see in the technique is of the micropropagation has been used to introduce variation in the offspring. Reason, it is not possible to generate virus free plants by micropropagation. Both assertion and reason seems to be the false over here. The reason why, because micropropagation used for rapid vegetative multiplication of the plants, and as the size of the propagules is minute, thus the technique is named micropropagation. Each such plant will be genetically identical to the parent plant. Generally apical or axillary meristems are free from the virus, hence can be used as explants and tissue culture to produce virus free plants. So, option 4 is the correct answer for 140. Question number 141. Somatic hybridization can be done by protoplast fusion. So, question number 141. Protoplast fusion. So, it is for the somatic hybridization. Protoplast fusion can be done. So, option 1 is the correct answer. 142. Read the following 4 statements. You could see A to D. How many of the above statements are wrong? You could see like 3 statements are wrong in this case, like 142. So, in 142 you could see it seems to be like the 4 statements seems to be wrong in this case. 142. Chikungunya is caused by chikungunya virus. Colostrum is the first breast milk of mother which contains antibody and which protects the infant of the age of 3 months. Beer is manufactured by fermentation of barley malt by yeast species. Tissue culture can be used to obtain virus-free healthy plants from the diseased plants. So, option 2 is the correct answer. So, 3 statements seems to be incorrect here. So, question 143. Undifferentiated mass of plant cells grown on nutrient medium, that we call it as a callus. So, we have a callus. Question number 143. Callus is an irregular. So, it is an irregular and organized. Usually, the callus is a irregular and unorganized and undifferentiated mass of cells that you could see. Undifferentiated mass of actively dividing cells grown on the solid medium which gel by the agar. So, the medium of ordinarily contains the auxins 2,4-D and are often a cytokines like BAP. Both are growth regulators. So, callus is obtained within 2 to 3 weeks. So, for this question, option 1 is the correct answer. 144. 144, mule is an example for interspecific hybridization. So, 144, mule. Mule is an example for interspecific hybridization. Interspecific hybridization. So, 2 is the correct answer. 145, Isardale that we discussed out in the previous part. Isardale is obtained by 145. 145, Isardale. Isardale is obtained by Bicanary ewes and merino rams. So, option 4 is the correct answer. Question number 146. Pebrine in silkworm is caused by nosema. Nosema is the correct answer. So, option 3. 147. Pebrine in silkworm is caused by nosema. So, 146, 3 is the correct answer. 147. The scientific name of the moth which produces tassar is Anthrea millita. Anthrea millita is the correct answer for 147. Okay, then we move on to the next question, 148. For this question, Nosema bombicus which causes pebrine in silkworm is a protozoan. Protozoan, so option 2 is the correct answer. 149. Resistance to yellow mosaic virus in a Habilmoschus esculentus was transferred from the wild species and resulted in a new variety called as Parbani Kranti. This we already discussed, Parbani Kranti, yellow mosaic virus. So, this is a repeated question. So, question number 149. 150. The principal cereal crop of India is a rice. So, 150. The patterns also we have discussed, right? The rice is the principal wheat crop of India. Question number 151. The International Rice Research Institute, that is like IRRI, 151, IRRI, where it is located. Okay, for question number 151, it's in the Philippines. Mostly you go for India, it's not in India, it's in Philippines. Okay, so 2 is the correct answer. Question number 152. A protoplast is a cell without a cell wall because of protoplasm. So, protoplast is a cell without a cell wall. So, 152. 152, protoplast cell which has its cell wall removed by the mechanical enzymatic means. 153. In plant tissue culture, the callus tissues can be regenerated into complete plantlets primarily by altering the concentration of hormones, by the hormones. So, option 154 question. 153, 4 is the correct answer. Question number 154, totipotency. We discussed already the totipotency before the part itself. The totipotency means development of an organ from the cell in a cultural medium. So, development of an organ. So, option of question number 154, the capacity to generate a whole plant from any part out of the growth and in a test tube in a sterile condition, special media is called totipotency. So, 3 is the correct answer. Question number 155, assertion and reason. The honeybee queen copulates only once in her lifetime. Reason, the honeybee queen can lay fertilized as well as unfertilized egg. Both assertion and reason are true, but reason is not the correct explanation for the assertion, because queen is only fertile female in its beehive, having immense developed ovaries. She lives for several successive years, about 2000 or more years a day. The queen has a remarkable capacity of controlling the fertilization of its eggs. A fertilized egg is laid in a worker or queen cell, while an unfertilized egg in a drone cell, the later develops parthenogenetically. The queen mates only once in a lifetime. The sperm stored in a spermathecal fertilized her eggs as long as she lives. So, option 150, question 155, option 2 is the correct answer for this question. So, question number 156, the Nobel Prize that you could see, the Nobel Prize for the communication, the Nobel Prize for the communication, the study for the communication methods of honeybees was given to 156 question. It's Carl von Frisch. Honeybees, despite their small size, are one of the most sophisticated communication, where you could see in the series of classic experiments dating from the 1920s, the Australian biologist, you could see the Australian biologist here, where the person named biologist Carl von Frisch, This person, the Carl von Frisch, investigated how honeybees communicate to extend other, and the scientist extended works. It is the Nobel Prize with Lawrence and Tinbergen for its achievements in comparative behavioral physiology and pioneering works in communication between the insects. We usually call it by means of the waggle dance. So, the type of dance, it's a waggle dance, the prominent feature that you could see, right. So, for this question 156, 2 is the correct answer. Question number 157, Amrit Mahal is an drought breed. So, when you see this options, one out of this, like the drought breed, because it's a very good breed, Amrit Mahal. For drought prone areas, it can survive on dry grass if needed. It is known to be power and endurance. It was developed by the ruler of the Mysore state between 1572 to 1636 AD, mainly for the transportation of army equipment. Okay, so option 4 is the correct answer. Question number 158, which of the following are the statements above the breeding is wrong? By breeding, pure lines cannot be, by inbreeding, pure lines cannot be evolved. So, usually by inbreeding, pure lines will be evolved. So, question number 158, we can go the same question. 159, which of the following is an exotic carp species? Cyprinus carpio, 159, 2 is the correct answer. Okay, 160, which of the following diseases caused by Nosema bombylicus in a mulberry? In case of mulberry plants that you could see, the mulberry silkworm, pebrine. So, question 160, pebrine is the correct answer. Pebrine is the correct answer for question number 160. Question number 161, which is correctly matched apiculture with the honeybee? So, 161, we go with the option 3. 162, match the column 1 and 2 and select the correct options from the codes given below. You could see out of the 4 options, A with 3, B with 4, C, that is wheat with emigre, and cowpea with the Pusa savani. So, option 1 is the correct answer for question 162. Question number 163. 163, during the process of artificial hybridization, if the female plant bears bisexual flowers, then emasculation is done with the help of forceps, that is the removal of anthers. The removal of anthers which usually takes place, this we already discussed the before part itself, right. So, the emasculation and bagging, these are all the techniques that we discussed. Removal of anthers from the flower bud before anther dehiscence, so that our desired anther will be dusted onto the surface of the stigma. So, question number 163, 3 is the correct answer. 164, Pusa komal varieties in cowpea is resistance to bacterial blight. So, option 4 is the correct answer for question number 164. 164, for this question 4, that is bacterial blight is the correct answer, because bacterial blight, where you could see the cowpea has been developed by hybridization of P85-2 and P426 cowpea strains. It is widely adapted, bears good quality pods, resistance of bacterial blight, and gives higher pod yield. 165, aim of plant breeding is to produce all of this actually. So, all the 3 options, disease resistant varieties, high yielding varieties, early maturing varieties. So, all the options seems to be correct for 165. Then we move on to 166, identify a macroorganisms that can produce the biomass of a protein. For this particular question, the biomass of protein, when you see the methylophilus methylotrophus, where you could seems to be the correct answer. Methylophilus methylotrophus seems to be the correct answer. First option is the correct answer. So, methylophilus methylotrophus. 166, one is the correct answer. Question number 167. The phenomenon that operates in the formation of root or shoot in a callus culture is differentiation. So, 167, you could see differentiation. So, differentiation, this is for the callus. 167, that was 160 differentiation. 168, tissue culture technique can produce indefinite number of plants from the small parental tissue. The economic importance of technique is called as genetically uniform population identical to the original plant. So, option 1 is the correct answer for question number 168. Micropropagation is used for rapid vegetative multiplication of plants as the size of the propagate is minute. We discussed already about this one. So, 1 is the correct answer. 169, somatic embryo can develop in plant tissue culture from the somatic cell. So, 169, 1 is the correct answer. 170, the breeding of unrelated animals, which means of individuals of same breed or between different breeds of different species, known as outbreeding. So, outbreeding is the correct answer for this particular question. This is for question number 171. Question number 172. Coming to 172, the place the Haliker dwarf breed of cattle is found in Karnataka. So, question number 172, Haliker you could see in Karnataka. This you could see in case of Karnataka. Okay. So, we are done with this part. Then 173, which of the following poultry breeds is not an English breed? Minorca, Australorp, Success, Orpingtons is all like an English breed. 4 is the correct answer. Question number 174. Percentage composition of fibrin and sericin in silk is 80:20. Option 2 is the correct answer. 175. The number of chromosomes present in the cells of the bread wheat Triticum aestivum suggests that it is exoploid. Answer 1 is the correct answer. 176. Triticle is developed through the intergenic hybridization. This we already discussed with this part. So, this is a question number 176 also, because a triticle which is between the rye and the wheat we discussed. 177. Maize hybrids have been developed for higher amount of lysine, because lysine rice varieties of maize, that is Shakti, Ratan, Econoproteina, have been developed in India. So, 1 is the correct answer. 178. Plants obtained through tissue culture are genetically identical and they are obtained by somatic cells. What do you call them? We call it as a soma clone. So, option 2 is the correct answer. Question 179. Totipotency is the best principle, the basic principles of tissue culture. So, going for the last question, that is like 179, totipotency. So, totipotency is for the tissue culture. This is for the tissue culture. Okay, so we are done with this part, the tissue culture. The last question, pollen grains of a plant whose 2N, the diploid, is 28 are cultured to get callus by tissue culture method. What will be the number of chromosomes in the cells of the callus? It means the number is 28 divided by 2. So, 180. So, 28 divided by 2, this leads to gives to the 14. So, this is the question number 180. So, by this we done with this part. This question discussion is enhancement in food production. So, by this we are done. In spite of this, if you have any doubts or any queries that you want to discuss, like please feel free to ask us. I'll be happy in assisting you. By this we are completing this the paper discussion on this particular chapter. Thank you so much. Have a great day.